Genes, chromosomes, RNA, AQA Biology. Key terms. Gene, a small section of DNA which contains the coded information for making a polypeptide and functional RNA. Somatic cells. Normal body cells contain 46 chromosomes, 23 pairs. Diploid. Somatic cells of T sets containing 46 chromosomes. Haploid is gamete cells. It's one set of 23 chromosomes. Genetic code. How DNA codes for an amino acid. 3 bases code for each amino acid. Start codon signify the start of a coded sequence. Intron don't code for any of the amino acids in the final protein. Exon, the coding region. Histone, very long DNA molecules. The substance that is wrapped around. Homologous pair, a pair of chromosomes in a body cell made through mitosis. Alleles, different versions of the same gene options. Human Genome Project, a project which mapped all of the human genes' location in every single gene of DNA. Protein synthesis is transcription and translation. mRNA, made during transcription. mRNA is a single polypeptide strand. In mRNA is a group of 3 adjacent bases called a codon. Genes and the genetic code. So what is special about the mitochondria and chloroplast? DNA code contains coded information for a polypeptide and functional RNA. This is due to evolution. Your mitochondria you get from your mum. And the disadvantage of mitochondria mutations is that they're passed on to future generations. So if your mum has a mutation, your children are going to have mutation and so on and so on. Scientific reasonings for the genetic code. Well, there are 20 different amino acids, and each amino acid must have its own code of DNA. And as there's 4 bases in DNA and 20 have to be coded for, if it was just 4 to the power of 1 is only be 4 that would be coded for. If there was just 4 to the power of 2, then only 16 would be coded for. So you'd have 64 chains of amino acids due to the 4 to the power of 3, which codes for 64 different substances. However, only 20 of those are needed for the 20 amino acids, which means that different codes are degenerate. So what's the purpose of the other 44 chromosomes? Because remember, only 20 of them are needed for amino acids. Well, you have stop codons, and some of them are spare, so they have more than one code, which is what degenerate means. So the features of the other 44 codes, sorry. Few amino acids are only coded by a single triplet, so it must have a degenerate code, which are coded for by more than one triplet. They're always read in the direction 5' to 3'. They contain start codons, stop codons, and the code is non-overlapping and universal. So it's the same in all of the different organisms of the same species. No, it's universal. It's all of the species. Sorry. Changing in one basis results in enzymes becoming non-functional as the sequences change. The primary sequences change, which means hydrogen bonding, ionic bonding won't bond in the same place. So due to this, the tertiary structure and the quaternary structure isn't the same as what it would have been. Base sequences in gene changes. It may code for a different sequence of amino acids, so it might form for a completely different protein, or might not form anything at all. This is because the code could be degenerate, so you might have, let's say you have AGG, and instead you have AGA, and because the code is degenerate, it might still code for, I don't know, lysine. Let's make up, I don't know. But yeah. DNA and chromosomes. How DNA is packed into chromosomes. So you have your DNA molecule, which binds with histones. The DNA histone complex is coiled, and the DNA histone complex is actually called chromatin. And the loops coil and pack together to form chromosomes. So what do genes do? is a sequence of nucleotides at a fixed position on a strand of DNA that specifically codes for a sequence of amino acids that form a primary structure of a protein. The genes contain exons and introns, which are coding and non-coding DNA. The sequence of bases in the gene specifies the amino acids that will be coded for. The code for the protein that is specialized by the DNA has been carried to the ribosome so they can assemble the correct sequence and produce the right protein. Structure of ribonucleic acid. The location is in the cytoplasm. And RNA polymerase codes for the complementary base pairs. Thymine becomes uracil and joins complementary RNA nucleotides. And this is just a little diagram of what RNA looks like. Now, this is all there is for this topic so far. And the next unit will be about translation and transcription and genetic coding. So if you like this video, then please like and subscribe, and I'll see you in the next one.